Okay. Good afternoon, ladies and gentlemen. My name's Tiernan. I'm a PhD student and also a trainee surgeon. And I'm going to be talking to you this afternoon about lighting up bowel cancer. So I'll first of all give you some background to the problem, what the actual clinical problem is that we set out to solve, my proposed solution, and then I'll conclude and take some questions. So colorectal cancer, or bowel cancer, it's more commonly known, is incredibly common, and these numbers still always surprise me. So 1 million cases every year worldwide, 40,000 new diagnoses every year in the UK alone, and the majority of these undergo a major operation. Now, something I noticed very early on in my training as a surgeon was that when something goes wrong, and things do go wrong, it's major surgery, they're pretty devastating. So almost 7,000 patients each year will have a major complication. This can result in a permanent colostomy bag, long stays on ITU, like in this, sorry. I'll just go back. Long stays on ITU, like in this picture, and actually 1,200 people every year will die as a direct result of complications, and that's not from the cancer itself. So what do we do for bowel cancer at the moment? Well, surgery is the only way you can cure it, and it's actually based on a paper here by a group in Leeds from over 100 years ago, and they showed that to try and cure a tumour, you don't just take the tumour, you also need to take out the lymph nodes, and this is a sort of network of channels, a bit like blood vessels but smaller, and they drain fluid from all over the body, and that's where cancers tend to spread first. So you need to take them out to work out whether it's spread or not. So it's very much a one-size-fits-all approach. So any sort of bowel cancer, however advanced really, we tend to do the same sort of operation and have done for a long time. Now this is a specimen, so this is what's been taken out of a patient with cancer. That's where the tumour is. This is the bowel, and as you can see we take away quite a lot of normal bowel as well. This bit in the middle is called the mesentery. This is basically a fatty layer that connects the blood vessels and the lymph nodes to the bowel itself. And in that are blood vessels and lymph nodes. This all needs to come out. So on a diagrammatic representation, if you have a tumour there, which is a common place to get one, that's the operation we do. We take that amount away, we join the two bits together. Now that includes those blood vessels and lymph nodes. But the problem is, about 60% of all these patients don't have any advanced disease. So they've got a tumour like that black spot, but it's not spread to the green lymph nodes. Now this means that they're actually doing too much for them, having a bigger operation than they need and potentially at risk of complications. So they probably only need that. So a smaller operation, shorter stay in hospital, and a lower risk of complications. Conversely, those who do have disease in the nodes probably need a bigger operation. Some work here in Leeds in the last few years and in Germany has shown that these patients have a lower chance of the tumour coming back afterwards if we do this bigger operation. So the problem is that we're over-treating those 60% of patients who don't have any nodal disease, as in it's just one single tumour isolated, and we're under-treating those patients who have got more disease than that, where it's spread to the nodes. So my solution I proposed is based around the fact that we do a lot of keyhole surgery now. So this has taken off in the last 10 to 15 years, and most cases are now done in keyhole. And in keyhole, you have a camera source that goes into the tummy and a television screen in theatre. So what I want to be able to do is inject something that glows, so a fluorescent substance that sticks to the cancer cells only, and then we can see it live in theatre on the screen. And by working out where the tumour is, we can then choose which of those 3 types of operation to do. So it's basically personalised surgery. Now fluorescence, I'm sure we've all heard of. So we have a fluorescent dye or substance or molecule, and if a certain type of light shines at it, it absorbs the light and then emits it again at a slightly different type of light, and it gives this fluorescent effect, which we can detect. And we can incorporate this very, very basically really into the camera system in keyhole surgery. Now sorry for the gory picture, this is the only one I've got. I was inspired for this project by brain surgeons. Now they use a different type of fluorescence, something that occurs naturally in a certain type of brain tumour. Now this is, I'm not sure if this pointer's working very well. You can't really see it, but that's a brain. This area down the bottom left is where they've removed the tumour. Now you can't see where the tumour is. It's very hard in brain surgery because you don't want to damage normal tissue and give someone a problem with function. However, when they shine a certain light at it, you can see this area up the top that's purple or pink. That's actually tumour and wouldn't have been resected without this technology. So my project involves making a fluorescent nanoparticle, which I'll come to in a minute, and then making it stick to bowel cancer cells only. Nanoparticles you may have heard about, they've become quite popular in the news over the last sort of 5-10 years. They're very small things, 100 nanometers, that doesn't mean much. That's 1,000 times smaller than the tip of a human hair, so tiny. The advantage is, if we inject them, they can pass out of the blood vessels and reach a tumour, which is why I'm interested in them. And I can also make them fluorescent. So after lots of trial and error, lots of different strategies and comparing different methods, this is what I've made. Now in the centre, that red bit, that's my fluorescent core. That's thousands of molecules of a fluorescent dye. Around that is some silica shell. And a silica shell is see-through, a bit like glass really. It protects the fluorescent core and stops it breaking down, and yet the fluorescence can still pass through it. Then I've got the yellow web on the outside is a chemical linker. I tried 4 different linkers as part of my work. This was the best. And the blue bit is a bit that only sticks to cancer cells. And that's what it looks like under electron microscope on the left. So that's a powerful microscope. And in the lab itself, that's a week's worth of work to make that little pile of powder. Now I wanted to design how to make it stick to the cancer. So cancer has lots of molecules on the surface. Different cancers have different molecules. Some molecules are also on normal cells, so it's very hard to pick which one to aim for. So I used 280 patients' samples, whether it's tumour and normal tissue. I looked at them under a microscope and analysed them to see which molecule on the surface that I could aim for would be best for this process. Now, to put this simply, these are slides from a microscope of cells under the microscope. The top are normal samples of bowel, the bottom 4 are tumours, and the 4 names at the top are these molecules that I'm testing. Basically, I need something that's expressed or that's present on the surface of all the tumour cells and none of the normal cells. That's the ideal. And CEA, this first column, comes pretty close. So it's very brown, meaning all the cells have it, and the normal sample is no brown at all, meaning they're not there. And when you analyse all those 280 patients, you find 94% of the cancers, this is present in a high level, and only 3% of normal samples. So this was by far and away the best one. And we published this work in the British Journal of Cancer this year. So that was our target. So we aim our particle and make it stick to CEA only, and that's on the tumour cell. To see if it works, once I've made the particle, you can grow cancer cells in the lab. So there's about 5 to 10 commonly used types of cancer cell that we can grow in a lab on a microscope slide, and that's what I've got there. That's the slide, and that grey bit is the cancer cell. Then we use a special microscope that can detect fluorescence, and I put my particle, my nanoparticle that's fluorescent and sticks to cancer cells, on this, then wash it off. And the idea is that it sticks to the cancer cells. So we use this microscope, which takes very special images and basically gives you an idea of an exact slice through these cells to give a picture like that. So for this cell analysis, what I found was, with my nanoparticle, when I stick it onto these cells and then wash off again, you can see that red fluorescent area around the outside. That's it sticking. So it works. But I wanted to make sure this wasn't a fluke and it was sticking because I designed it to do that. So I also made a dummy nanoparticle that isn't designed to stick to the particle, to the cancer cells, but otherwise is exactly the same. And this didn't stick at all. I then repeated this on 2 other types of cancer cell that we grow in the lab, so 3, specially selected to represent real human cancer cells. And I got the same results. Lots of fluorescence with my nanoparticle, very little or none at all with the dummy. Same on the 3rd one. Now this is quite unique because most nanoparticle studies on other cancers where they've tried to do this, they never show you the dummy nanoparticle because they always get this non-specific binding where it sticks anyway despite what you're aiming for. So we're pretty pleased with that. And we analysed lots of cells. We found that it's over 10 times brighter for the one type, 9 times brighter for another, and over 3 times brighter for another. And these cell types are known to have CEA on, so I know I'm targeting the right thing. And for the last bit, I designed a complex cellular system. So this isn't just cancer cells, all types of cell. So much more realistic of what it'd be like in a human patient. They're very notoriously hard to actually make these work, but I managed to get 4 with my particle, and 3 of them glowed brightly. And with the dummy nanoparticle, 3 systems we managed to make, and none of them glowed at all. So in conclusion, my particle works basically. It sticks to colorectal cancer cells only, and it glows. It's unique because it's never been done before in a colorectal cancer or bowel cancer model at all. And as I said, the vast majority of studies don't use this sort of controlled dummy particle to prove that it's working properly. So what does this mean for patients? Well, for those 60% of patients with the earlier tumours who are currently getting a major operation and put at risk of all these complications and death, It means they can have a smaller operation, less chance of getting a complication, and reduce their hospital stay. And for patients with the more advanced disease, we can select them out for the bigger operation and hopefully increase their chance of survival, as in reduce the chance of it coming back. So for the future, I'm currently writing a manuscript to publish all this work. We've aimed to do the first human trials using it here in Leeds. There'll be cost savings to the NHS because all these patients will have smaller operations and hopefully experience fewer complications. And I think probably the biggest potential of this is it could be applied to other cancers. Cancer surgery uses the same sort of paradigm and methods for resection. So in terms of removing lymph nodes, the blood vessels that supply the tumour. And it would benefit all these, prostate, breast, stomach, bladder and brain, all common cancers. The same principles could be used. The only thing you'd change is what molecule you aim for on the cancer surface to make these stick to. So I'd like to thank Cancer Research UK who funded this project for me for the last 3 years, and my supervisors. And I'll take any questions now. Thank you.